Good afternoon, everyone. My name is Xuejin. I came from Georgia Carnegie Group at the Brown University. As George has another conference on the fluctuation PDE, so he couldn't come here today, and he asked me to show some simulation results in our group. Today, I will talk something about the sickle hemoglobin self-assembly inside a red blood cell. In this talk, first I will give a brief introduction on sickle cell anemia, and then I will describe the coarse-grained sickle hemoglobin model we used in this study. Next, I will show you some simulation results on the self-assembly of sickle hemoglobin and possible shape deformation of red blood cell induced by the growth of sickle hemoglobin polymers. Finally, I also want to show some initial results with our new two-component red blood cell model. Okay, let's go into the first part. The sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecules in the blood. And the disease gets its name from the shape of red blood cells and a certain condition. Here, I would like to show you an interesting movie about the sickle cell anemia from Dowland DNA Learning Center. When the red blood cell travels in the microcirculation, some red blood cell inside the vessel become sickle shaped, and the elongated cell can get stuck in small blood vessels. So part of the body couldn't get the oxygen they need. The sickle cell anemia is caused by one single code letter change in the DNA, and this affects one of the amino acids in the hemoglobin and makes the hemoglobin molecules stick together and forming long fibers. And the long fibers distort the red blood cell shape and bring on attack. At the same time, the mechanical and rheological properties of red blood cell can be changed. From a molecule level, the packaging of a very high concentration of hemoglobin into red blood cells requires that the hemoglobin protein be extremely soluble. And a normal red blood cell has hemoglobin A that has 2 alpha and 2 beta subunits. When the red blood cells traverse in the microcirculation, the oxygen is released from the oxyhemoglobin and generates the deoxyhemoglobin. Upon deoxygenation, the replacement of a beta subunit at the site of beta 6 with valine results in a hydrophobic interaction. And this makes the aggregation of sickle hemoglobin into large polymer fibers. So the hydrophobic interaction is necessary for the formation of polymer fibers. And in the left bottom of this figure, a high-resolution model shows the interaction between deoxyhemoglobin from 2 different strands. In this figure, the alpha subunits are yellow-gray, and the beta subunits are gray. And the heme group are shown as the red spheres, and the blue and the green color represents the contact between 2 beta 6 sites. And the polymerization of the deoxyhemoglobin and the alignment of fibers result in a distortion of the shape of the red blood cell, and also a marked decrease in the deformability of red blood cell due to the increase of shear stiffness and adhesion of red blood cells. So in this study, we want to simulate the self-assembly of sickle hemoglobin and observe possible shape deformation of red blood cell induced by the growth of sickle hemoglobin fibers. In this study, we use the dissipative dynamic method to simulate the sickle hemoglobin polymerization. The dissipative particle dynamic method is the particle-based simulation method that models seamlessly soft matter and solvent and can correctly represent the hydrodynamic interaction in the system. In a standard dissipative particle dynamic simulation, it includes conservative, dissipative, and random forces. And the dissipative and random forces form DBD summer state. And for a DBD simulation, the force field includes two main parts, the bounded and non-bounded potential terms. In our study, the bounded interaction includes the hooking springs, the bound bending interaction, and the bending finitely extensible nonlinear elastic model interactions. And also, we use the hooking springs between two constitutive DBD particle and the bound bending interaction among three consecutive DBD particles to control the chain rigidity. And we also found that the molecular chirality can be used to drive the phase transformation from two-dimensional membrane to one-dimensional fibers. So in our model, we also consider the chain chirality in our system. And we use the bound bending interaction and the bending FENE interaction to control the chain chirality in our model. For the non-bounded interaction, we use traditional DBD conservative interaction. The red blood cell in this study is simulated by a multi-scale red blood cell model developed by our group. In the multi-scale red blood cell model, the membrane of red blood cell is modeled as a two-dimensional triangular network. We have several hundred DBD particles in this two-dimensional network. And the connection between two DBD particles are linked by a viscoelastic bond to impose proper membrane mechanics. And also, we consider the bending energy between neighbor triangles to describe the bending resistance of lipid bilayer. Also, we keep the surface area and the volume of red blood cell constant. Okay, next I will introduce the coarse-grid model of sickle hemoglobin. As we have known that one normal red blood cell contains hemoglobin A that has two alpha subunits and two beta subunits. So in our coarse-grid model, we use 4 DBD particles to represent one sickle hemoglobin molecule. And the packaging of a very high concentration hemoglobin into red blood cells requires that the protein be soluble. So we also consider the cytosol in our model, and a change from a charged to neutral hydrophobic amino acid causes aggregation upon deacidination. So while we let the yellow particle are hydrophobic, while the green particle are hydrophilic. Specifically, we let the green A particle represent the alpha subunit, and the yellow B particle represents the beta subunit. And the connection between 1A and 1B particle form 1AB dimer. And one sickle hemoglobin molecule is a tetramer formed from 2 AB dimers. And then several sickle hemoglobin molecules are connected each other to form a sickle hemoglobin single strand. After that, one of the two hydrophobic particles in one single strand can bind to another hydrophobic particle on partner strand to form a hydrophobic contact. So we can get a sickle hemoglobin double strand. And also a bond bending interaction is also included for the hydrophobic particle with their free end has a closed contact. Okay, then we simulated the assembly of sickle hemoglobin in a three-dimensional periodic system. And without the chain chirality among the hydrophilic and hydrophobic particle, we only find the small aggregation from the simulation. One possible reason is that the hydrophilic particle in this case can rotate freely in the system. So the coverage of hydrophilic particle on the surface of hydrophobic particle can avoid the direct connection between the hydrophobic particle and the cytosol particle. When we include the chain chirality, we get the elongated shade-like microstructure from our simulation. And the formation of this microstructure is a result of the local packing constraint among the hydrophilic particle and the hydrophobic particle due to the strong chiral interaction. Two DPD particles, one is hydrophilic and another is hydrophobic, are collinear with another two DPD particles on another strand. So the sickle hemoglobin pack parallel to each other to form a monolayer membrane to reduce the system energy. By decreasing the chain chirality, we can find that the elongated step-like microstructure from the sickle hemoglobin polymerization. And in this case, the hydrophilic particles are still restricted due to the chain chirality. So the hydrophobic particle had to pack more densely and to form cylinder microstructure to minimize contact with the cytosol particle. This can result in an aggregation of the sickle hemoglobin and the persistent growth of the sickle hemoglobin molecule in one dimension and to form the elongated step-like microstructure. And it has been demonstrated that the confinement has an influence on the self-assembly microstructure of biopolymer and soft matter. And the self-assembly of sickle hemoglobin inside the red blood cell is also one kind of confined system. So next, we want to observe the confinement effect on microstructure formation. So we simulate the self-assembly of sickle hemoglobin in bounded domain. And in this study, the confinement effect is realized by placing the sickle hemoglobin in a cuboid box confined in two-dimensional and three-dimensional space. From the simulation, we find that the elongated fibers and the network microstructure can be obtained from the sickle hemoglobin polymerization. And also, the type of the domain, the type of the boundary domain may also affect the self-assembly microstructure. So we also want to check the effect of the aspect ratio of the geometry on the self-assembly of sickle hemoglobin. To this end, we put the sickle hemoglobin in a cuboid box. So the one dimension has a strong confinement. So the confinement behavior is more pronounced in this strong confinement direction. And from the simulation, we also find the elongated fibers. So next, we want to study the self-assembly of sickle hemoglobin inside a red blood cell. We first simulate the self-assembly of sickle hemoglobin inside a rigid red blood cell by freezing the red blood cell membrane. And we get the elongated polymer fibers through the simulation. However, several elongated fibers buckle to conform the red blood cell membrane curvature near the boundary. In this case, the red blood cell is rigid, and the shape of the red blood cell membrane can be changed. So it's reasonable that the shape of the red blood cell can affect the self-assembly microstructure. But in the biological system, such as red blood cell, the self-assembly takes place in a confinement environment with a soft boundary. So the self-assembly microstructure should be different because of the flexible boundary. Here we show the response of red blood cell obtained due to the aggregation of sickle hemoglobin. We find that when we include the cytosol particle in the red blood cell, and we found the red blood cell can keep its shape. But the strong red blood cell membrane fluctuation takes place when we include the sickle hemoglobin molecules in the red blood cells. To give a quantitative insight into some fluctuation of red blood cell, we compute the membrane fluctuation distribution from the simulation. And in the right below figure, we show the difference. We find that the red blood cell with sickle hemoglobin has a wide distribution. This means that the red blood cell has a strong deformable with the self-assembly of sickle hemoglobin. Actually, it's difficult to simulate the self-assembly of sickle hemoglobin inside the red blood cell because of the moving and elastic boundary of the red blood cell membrane. And for a strongly fluctuating red blood cell membrane, and the impenetrability of between the red blood cell and the sickle hemoglobin become important and lead to the strong repulsion between them. And this also affects the self-assembly of sickle hemoglobin. So in our study, we didn't find the self-assembly microstructure from the sickle hemoglobin inside the red blood cell. To keep away from this difficulty, we can start the simulation by placing an elongated polymer fiber inside a red blood cell and use a linear swim model to simulate the growth of sickle hemoglobin fiber. Indeed, when we increase the bond length of the sickle hemoglobin fiber, we find the sickle hemoglobin can deform the red blood cell shape, and the red blood cell can become sickle-shaped. Here I will show you a movie show the dynamic pathway. And also in the experimental observation, the red blood cell membrane is penetrated and destroyed by the formation of sickle hemoglobin fibers. So the cytoskeleton can dissociate with the lipid bilayer, but we couldn't find this phenomenon with our single component red blood cell model. Our group developed a new model, and in this new two-component red blood cell model, we use two different components. One component is the lipid bilayer, and the other component is cytoskeleton. And each component is composed of two-dimensional triangulated network. different from the single component red blood cell model. There, for the lipid bilayer component, there is no shear stiffness, but only has bending stiffness and a large local area stiffness. And for the cytoskeleton, there is no bending stiffness, but possesses shear stiffness. And also we include the elastic and the friction interaction between the lipid bilayer and the cytoskeleton. And then we test our new two-component red blood cell model by using the sickle cell polymerization. We find that the growing sickle hemoglobin fibers can push the red blood cell membrane and lead to uncoupling between the lipid bilayer and the cytoskeleton. And this is agreement with the experimental result. Here I show the dynamic uncoupling pathway. And also we can track the growth rate of polymer fiber during the uncoupling process. Okay, I will conclude my presentation. First, through the simulation of self-assembly of sickle hemoglobin, we demonstrated that the hydrophobic interaction is necessary for the formation of sickle hemoglobin fibers. And the chain chirality is the main driver for the sickle hemoglobin polymerization, and the self-assembled elongated fiber can be obtained in hard confinement and also the open space system. But we can't find these elongated fibers in red blood cell membrane with the soft boundary condition. But we can use the linear elongation and bending interaction of sickle hemoglobin fibers lead to the sickle-shaped cell. Last, I will give my thanks to my advisor, Professor George Kaniadakis and Bruce Caswell at Brown University. And also I want to give thanks to Dr. Zhang Lipeng from MIT. He developed the two-component red blood cell model. And also Huan Lei from Brown University, also Dmitry Fedosov. He was in our group, and he made a great effect on the multi-scale red blood cell model. He then moved to Germany and to build up his own group after he graduated from our group. And also, I would like to thank the financial support by the NIH and NSF grant program. Thank you for your attention.